Here we have a red blood cell, an erythrocyte, and we know that the A, B, O group antigens are on the surface of the red cell. And with the rhesus factor, it's exactly the same. The rhesus factor is an additional factor, and it's either present or absent on the red cells. So here we have a diagrammatic representation of the rhesus factor antigen. It's a protein, and in fact it may be a membrane transporter protein. There's currently debates about what the rhesus factor actually does in biochemical terms. But it's either present or absent. Now it's actually quite complicated. There are many subgroupings to the rhesus factor. The last time I checked there was about 49 subgroupings. But the main one, the important one, the antigenic one that we need to know about is the D factor. So when we're talking about the rhesus factor, we're really talking about the D factor. So it's important to realise how this factor comes to be present on the erythrocyte. And it is entirely genetic. It's transmitted by Mendelian inheritance. So the rhesus factor is coded for by a dominant gene. And the chromosome is number 1. So here we have a diagrammatic representation of the chromosome number 1s. We have a homologous pair of chromosomes. And the rhesus factor is going to occupy one of the positions on that chromosome. So this gene in this position contains the genetic information required to synthesize the proteinaceous rhesus factor. Now, a dominant gene is one which will be expressed if present. And the rhesus factor is coded for by a dominant gene. So by convention, we denote dominant genes as a capital letter, and the recessive gene, which would result in no rhesus factor being present, we represent with a lowercase r. Now, if there's rhesus factor incompatibility problems leading to hemolytic disease of the newborn, that always occurs with a rhesus negative mother. Now, in the UK, about 15% of the population are rhesus negative. So it would be reasonable to expect 15% of women to be rhesus negative. In Asian populations, Chinese and Bengalis, for example, it's as low as 1 to 5% of women that are going to be rhesus negative. But in the UK, it's about 15%. And we have a homologous pair of chromosomes with the relevant gene. So if the mother is rhesus negative, that means she does not have a rhesus positive gene. So we can know for sure if a woman is rhesus negative, or indeed if anyone is rhesus negative, they will have 2 small r's there. They will both be recessive genes. But if someone is rhesus positive, then one or both of these genes may be dominant. So if someone was a RR, they had 2 copies of the dominant gene, I think you can see that they would be rhesus positive. But if someone was RR with 1 dominant and 1 recessive gene, the gene for the production of the D rhesus factor is dominant, so it would be expressed if present. So that allows us to do some genetic calculations or genetic plans to decide the probability of rhesus factor incompatibility occurring, resulting in hemolytic disease of the newborn. Because this is only going to happen with a rhesus negative mother and a rhesus positive father. So we know that the mother is going to be rhesus negative. She's going to be 2 small r's. And the father could be 1 large R and 1 small r. He could be carrying 1 copy of the rhesus gene, and the other position on the other homologous chromosome could be the recessive gene. In other words, this father is heterozygous for the trait, whereas the mother is clearly homozygous recessive. Now, to reproduce, the mother will make ova, the father will make sperm, and these are going to recombine. So one possibility is that combining with that, and that will give us a rhesus positive fetus. Alternatively, that could combine with that one, and that will give us a rhesus negative fetus. Alternatively, it could be that one with that one, resulting in a rhesus positive fetus, because there is one copy of the rhesus gene present, which of course is dominant. Or it could be that one combining with that one, which will give us two small r's. So here we see the probability is 50-50 that any particular fetus will be rhesus positive. And the presence or absence of the rhesus factor is entirely genetic. Now, alternatively, we know the mother's going to be rhesus negative. The father could be homozygous for the trait with two rhesus positive genes. Phenotypically, the father is still, of course, rhesus positive. In this case, the mother produces ova, the father produces sperm. So this could combine with that one, giving us Ra. Because there is one copy of the rhesus positive gene, that is going to give us a rhesus positive fetus. Likewise with this possible combination. In fact, likewise with that one, and indeed likewise with that one. So in this case, even though the mother is rhesus negative, 100% of potential fetuses formed by this union would be rhesus positive. Therefore, the probability of rhesus factor incompatibility occurring is going to be high. So the rhesus factor, really quite simple. It's either there or it's not. It's coded for by a single dominant gene, which may be present in homozygous or heterozygous form in the father. So consider rhesus factor incompatibility whenever we have a rhesus negative mother and a rhesus positive father.